So hello Hasvora audience, welcome back. Today we'll be studying another paper of this is the expected paper of 9700, the biology practical of for the October-November 2025 session. Time now for this paper is 2 hours, but in this video we will only be covering the question 1 that is expected. Okay, for the question 2 you already know much stuff about that, and also I have covered all of the stuff related to the question ones and the question two on my channel, so you can just visit it. I have just dropped the link in the description down below here where you can find all of the videos for the plan diagrams, for the higher magnification drawings. Okay, all of the types have been covered. Okay, so this is the expected paper for the question number 1. Okay, so without further ado, let's start because I don't want your brain cells to suffer more. Okay, so this question is actually for the diffusion, and this is actually for the dialysis tubing. Okay, now these type of questions are not much tested, but this time there is a chance that they can be tested. Okay, so this is for the rate of diffusion question. Okay, and let's just enclose it in this box. Okay, let's start. So before you proceed, read carefully through the whole question 1 and 2. Plan the use of 2 hours to make sure that you finish the whole of the question 1 and 2. So question number 1 says whisking tubing is a selectively permeable membrane. Okay, it is just like a cell surface membrane, so you can just treat it like it is actually the diffusion can occur through it. Okay, similar to cell membrane, so some biological molecules are able to diffuse through the whisking tubing membrane. You will investigate the effect of temperature on the diffusion of reducing sugar through the whisking tubing. Okay, so what we are doing is that we are actually checking the effect of the temperature on the diffusion. Okay, so you will need to prepare a cereal dilution of 10 percent reducing sugar R. Okay, so estimate the concentration of reducing sugar in the water at two different temperature. Okay, so now most of you will be wondering that what is the use of the cereal dilution here. Now remember that whenever actually sometimes you have seen like most of the time we have seen the experiment that we do the seal or the simple dilution, and then at the end of the experiment we are given solution P, or you can say something like that, solution S. Okay, and then we test for that as well. So the seal dilution is actually used to you can say reduce the concentration of that unknown solution. Okay, so here we do not know that how much of the reducing sugar will be diffused into the external water. Okay, or you can say down the potential gradient into the distilled water. So we do not know that how much of the reducing sugar will be diffused. So this is actually your unknown concentration. So we will be using that seal dilution to actually reduce the concentration of how much of the reducing sugar has moved into the water. And now for those who are actually were wondering that we haven't seen a tubing in our life, so I completely understand that most of the students do not most of the actually teachers do not cover this experiment because this is not usually tested. But just for the sake of that, so I'll be covering that how a whisking tubing is actually looks like and how you have to set it up, because most of the students are actually struggling to actually open and fill it with the solution. Okay, so this is how your whisking tubing actually looks like when it is just closed, or you can say it is not filled. Okay, it will be given to you in the examination, but it is just like just you can say a piece of tape. Okay, and most of you do not know how actually to open it, but I think the examiner has provided some information about that in the paper. So what you have to do is that you have to soak the tip of whisking tubing, okay, in distilled water. Okay, because you cannot open it like this. So You have to soak the tip of the whisking tubing, and also you have to rinse the whisking tubing, because in the case if there is solution that is already present on this outside of the whisking tubing, that will actually contaminate and obscure your readings. Okay, so you have to soak the tip of the whisking tubing and rinse it well, and then you have to gently rub the tip against your fingers. Okay, and once you do that, the tip will actually slightly open. Okay, and then what you have to do, you just have to fill the syringe with the required solution. In this case you have to fill it with the reducing sugar solution R. Okay, and then you just have to um, press the syringe into it, and you just have to fill it with the solution. It will automatically open like this. Okay, so this is how it looks like when it is filled. Okay, so I hope you understand. And just for the practical demonstration, I have also attached a video of how you open and fill a whisking tubing. Okay, so you can just watch it in the description down below. Okay, so now the examiner is saying that you are provided with the material shown in the table 1.1. So the solution R is equal to 10% of the reducing sugar solution, and the solution G is 20% reducing sugar solution. Okay, and then you have the Benedict solution. It is probably for the test you have to perform on that. Okay, and you have to check that what is the time taken for the color change, and then you have the distilled water. It is for when you have to fill the outside of the whisking tubing so that the diffusion can occur. Okay, and let me tell you one thing more, that the lower the concentration of the reducing sugar, the more time is taken for color change. Okay, you should know this. Okay, so in the case if a sample takes more time to show a color change, this means that it has lower concentration of reducing sugar. Okay, now let's move on. Now the V is actually your two lengths of 15 cm cube whisking tubing in a beaker of water. Okay, so they have already given you in a beaker of water because they know that most of students find it challenging, and it is actually not very obvious. Okay, in the case now, only those students who have done this experiment will know that it is a little bit challenging to open the whisking tubing for the first time. Okay, so I usually cover that type of challenges, or you can say the hard points of the questions, because I know that when the student will be actually performing that experiment, he will find it difficult. Okay, so then the P is the beaker containing water at room temperature. Okay, so P is actually your solution with the room temperature. So the other one will be you can say the Q, which is beaker containing hot water. Okay, so now these are your main two temperatures. Okay, this is the room temperature. What is the room temperature? Mostly it is 27 degrees Celsius, and this will be let's see what the examiner has provided. So now you will set up the apparatus as shown in figure 1.1 and using the step 1 to step 6. Okay, so tie a knot in one piece of whisking tubing as close as possible to the end. Okay, so what you have to do, the whisking tubing looks something like this. Okay, so you just have to tie a knot here. Okay, so one of the end is closed. Then uh, so then you have to open the other end, wet the whisking tubing. Okay, so and rub the tubing gently between your fingers. Okay, so just the method I just told you. Okay, and between your fingers and thumb, put 10 cm cube of G. Now what was G? G was 20% reducing sugar solution. Okay, it was 20% reducing sugar solution. Then we have to rinse the outside of the whisking tubing by dipping it into the water and the beaker label V. So V will be the distilled water, okay, and it will be actually for rinsing, not for adding into the beaker. So put the whisking tubing into the large test tube P1 and put this test tube into a test tube rack. Okay, so fold the open end of the test whisking tubing over the top of the large test tube. Use a paper clip to hold the whisking tubing in place as shown in figure 1.1. Now you might be given the paper clip, or you can also be given a tape to actually seal the you can say the open end, okay, with the wall of the test tube. So this is how your test tube will look like. Okay, so this whisking tubing will actually have the 20% of the reducing sugar solution, and then you will have this in the last test tube, which they have called P. Okay, this is actually your P, and not in the bottom of the whisking tube, and a paper clip to hold the whisking tube in the test tube. Now most probably you will be filling it, okay, with the distilled water, so diffusion can occur. Then you have to repeat step 1 to 6 with other piece of whisking tube and last test tube labeled Q1. Now the P was actually for the room temperature. Now this is for the beaker with the hot water, okay, so you just have to repeat it with that one. Then record temperature of the water present in beaker P. So beaker P was the one with the room temperature, so let's just assume that it is at the room temperature, and you will be given the thermometer for that, so it is 27 degrees Celsius. Then step 9, prepare a water bath at 60 degrees Celsius using the beaker Q. Okay, so beaker Q has the you can say the water at 60 degrees Celsius. So let's just mention it here. So it is containing hot water at 60 degrees Celsius. Okay, and the water may be need to be heated. Okay, so use a syringe to transfer distilled water W now into each of the last test tube containing the whisking tubing. Okay, so now you will transfer this distilled water into the test tube P and Q, and it will be actually here. Okay, it will be here. So distilled water will be here. So then the diffusion can occur. Okay, so the level of the water in each last test tube must be just above the level of G in the whisking tube. So for example, now if the G is still the level of here, so the distilled water should be till here, okay, just above it. So now they have given you the demonstration of that as well. Okay, so when you fill the water, it should be above the level of G. Then put the test tube P1 containing whisking tubing into the beaker P. Okay, now this is the one with the room temperature. This is the one with the room temperature. And put test tube Q1 containing whisking tubing into the beaker Q, and this is the one with the 60 degrees Celsius temperature. So start the timing and leave for at least 10 minutes. Okay, so why are we leaving for 10 minutes? So the diffusion can occur. Okay, and we are actually keeping the same time, so that we actually just keep all of the other factors constant while we just check for the temperature. So during this 10 minutes continue with step 14 to 18. So put 2 cm cube, so put So put 2 cm cube of distilled water W in each of the small test tube P2 and Q2. Draw a line on each test tube at the level of the water. Okay, so now you will be just putting 2 cm cube of the W into each of the small test tube P2 and Q2. Okay, now these are the another test tubes. Okay, these are not the same as the P1 and Q1. You have left that. Okay, so now you are just adding 2 cm cube of distilled water into P2 and Q2. Let's see that what the examiner wants us to use it for. So pour this water into container labeled for waste. Okay, so now we are just rinsing. Okay, we are just rinsing when we added 2 cm cube of distilled water. Okay, because then we added it to the container for waste. Okay, so it is mostly used for the rinsing. Then put the test tubes P2 and Q2 into test tube ready rack for step 21 and step 22. Okay, so now I have figured out that what we'll be using it for. So once the diffusion is complete, okay, once the diffusion is complete, this outside solution will have some sugar solution. Okay, this will have some reducing sugar solution, okay, because the diffusion would have occurred. Okay, so you will be moving this external solution into the for the P1, the external solution of the P1 into the P2, and the external solution of the Q1 into Q2. Okay, so this is the purpose for which these two other test tubes are used. Okay, because that one will actually have the whisking tube, and this one will be used to test at the end. Okay, not right now. Okay, so now what we are doing right now, you will now need to carry out a serial dilution. So serial dilution generally means that halving the each successive you can say the concentration at each successive uh, dilution. Okay, so for example, if you have 10%, you actually have to reduce it to 5%, then 2.5, then 1.25, then 0.625. Okay, like that. But here you have to reduce the concentration by a factor of 10. Okay, so just be vigilant that you read the question properly, because he can demand anything. So reduce the concentration by a factor of 10 between each successive dilution. You will need to prepare 4 concentration of reduced sugar in addition to the 10% of reducing sugar solution R. Okay, so for example, if I have 10%, so what will it be if I just reduce it by a factor of 10? Just divide by 10. Okay, so it will be 1%, then again it will become 0.1%, then again it will become 0.01%. Okay, so now I have three other solution in addition to the 10% and one more, so it will be 0.001%. Okay, so this will be your uh, concentration that you have to prepare. Now most of students will be wondering that whenever we are actually halving the concentration, we actually take half of that concentration and take half of the water. Okay, so what we'll be doing here, so if you cannot understand it like that, so one of the main basic idea is that you are actually reducing it by the factor of 10. You are actually taking 1/10 of it. Okay, so what you will be doing is that you will take 1/10, or you can say 1 cm cube here, because the total solution is of 10 cm cube. So you will be taking 1 cm cube of each concentration. For example, if I am preparing, like, you can say 1%, so I'll be taking 1 cm cube of this concentration, and the rest will be water. Okay, so what is the total? It is 10. If I take 1 cm cube of the 10%, this means I'll be adding 9 cm cube of distilled water W. And if you cannot understand like this, what you can do, you can just apply the formula. M1 V1 is equals to M2 V2. Now here, this M2 is the concentration that you want to prepare, the V2 is the total concentration that you want at the end, then M1 is the concentration that you are using, and V1 is the concentration that you have to use, okay, of that concentration. So for example, if I have the M1 which I'm using is 10%, I do not know how much volume I need to take out. Okay, so it is V1, and what concentration I need to prepare, it is 1%, and how much volume I need to prepare, I need to prepare 10 cm cube. So if you just take out V1, it will be 1. This means you have to take out only 1 cm cube of 10%, and the rest will be water. Okay, so this formula only tells you the volume you have to take out of that concentration that you are using. Okay, hope you understand this. So after the state dilution is completed, you will need to have 9 cm cube. Okay, look, now the exam is also giving you the hint. You will have 9 cm cube of each concentration available to use. Why 9 cm cube? Because you are transferring the 1 cm cube successively to the other one. Okay, that is why you will have 9 cm cube left behind. So complete figure 1.3 to show how you will prepare your seal dilution. Figure 1.3 shows your first two beakers you will use to make your seal dilution. You will need to draw 3 additional beakers. For each beaker, add labeled arrows to show the volume of reducing sugar solution transferred. So actually it will be 1%. Okay, so and the volume of the distilled water added. Okay, so it will be 9 cm cube. So under each beaker state the concentration. Okay, so now these are 3 easy marks, you just have to draw it. Okay, and I have showed multiple times how you have to do it, so let's just do it now. So you have 10 cm cube of 10% reduced sugar solution R in the beginning, and then you have 0 cm cube of water. And now what you have to do, you have to transfer 1 cm cube. Okay, so 1 cm cube of 10% reducing sugar solution. Okay, you have transferred that. This is the one that you have transferred. Okay, so and then what you have to do, you have to, let me just write it here. Then you have to transfer 9 cm cube of W, which is the distilled water. Okay, and then what will be the concentration if I do this? Okay, I have reduced it by the factor of 10, so you will write 9 cm cube. Why? Because 9 cm cube, because you have to transfer 1 cm cube from this as well. So 9 cm cube of 1% of reducing sugar solution to use. Okay, and then once again you have to transfer from this one. So you just have to transfer like this. Okay, let me just draw it correctly. You just have to draw it like this. Okay, and then we will have a beaker. Let me also draw a beaker right now. So you will have a beaker. Okay, and let's complete the other end of the beaker as well. So this will be our beaker, and then now what we are doing here, we are adding, let me just 1 cm cube of how much? 1% now. Okay, 1% reducing sugar solution. Okay, now you have added 1% of the 1 cm cube of 1% reducing sugar solution. Okay, and you will be adding 9 cm cube of water. Okay, so like this, and you will add, let's just reduce the size of this, you will add 9 cm cube of W. Okay, and then once again you have to write below this that what is the concentration that you have now. So it will be 9 cm cube of how much? This time you will have 0.1% of reducing sugar solution to use. Okay, so let's just copy this again. Let me just copy this. Okay, so let's just copy this and make it once more. So it will become 0.01%, and then once again okay, like this. So it is actually moving out of this plane. Let me just reduce the size of these. Okay, okay, so now it is perfect. Let's just also adjust this line. Okay, and then this will also be transferred like this, and let's move a little bit down this one. Okay, So this will also be transferred. Okay, so now let's just change the readings of this one. So the first one was 1%, this second one will be 0.1%. okay, this will be 0.1% reducing sugar solution, and 9 cm cube of W. So this will actually become 0.01%. So this will become 0. Uh, this will become 0.01% of reducing sugar solution to use, and then the next time we will have 0.01% solution. So it will be 0.01% solution that you are adding 1 cm cube of. Okay, and then what you will have to prepare, you have to prepare 0.001%. okay, so it will be 0.001 1% of reducing sugar solution to use. Okay, so this will be your whole diagram for 3 marks. Let's move on to the next steps. Okay, so now the examiner is telling you that you have to carry out steps 16 to 29. So prepare the concentration of the reducing sugar solution as decided. So we have prepared from 10% to then the 1%. okay, and then the 0.1%, then the 0.01%, and then the 0.001%. okay, so you have just prepared these all concentration. Label 5 small test tubes with the concentration you have prepared. So we will have to label that test tubes, and then put 2 cm cube. We have to put 2 cm cube of each concentration of reducing sugar solution into the approximately labeled test tube. okay, so this means, for example, if you have the test tube that is being labeled as 10%, so you have to put 2 cm cube of 10% into this. okay, so you have to put 2 cm cube of the each of the concentration into its respective test tube. okay, and then uh, you have to put these 5 test tubes into test tube rack ready for step 24. After at least 10 minutes, remove the test tube P1 and Q1. Now remember, we have left this for the diffusion. okay, now the diffusion will have been completed, so you just have to keep track of that, and now you have to remove it from the beakers and put them into the test tube. okay, because now you want the diffusion to just finish up. okay, and now you just want the sugar content that has been diffused to test for that. okay, but before that we'll be testing for the concentration that we made up. okay, because we'll be using that to compare. So remove the whisking tubing from the test tube P1 and the Q1, put these into the container labeled for waste. okay, so do not throw away, and the solution are remaining in the test tubes. okay, so because you have to use it for the testing. Use a pipette to transfer solution from last test tube P1 into a small test tube P2. Now you are saying that what we are doing is that we are using the P2 for the testing. okay, so up to the line you drew in the step 14, use the pipette to transfer from the last test tube Q1 into a small test tube Q2. okay, so you also be using to test that, and carefully remove some water from beaker Q so that there is approximately 250 cm cube of water in the beaker. Use this as a water bath and heat to the boiling ready for step 25, because now you will be performing the Benedict's test on each of the concentration that we prepared. okay, so put 2 cm cube of Benedict solution B into each of the 5 small test tubes. Now you are seeing that what the examiner is actually doing the same thing that I told you. So you'll actually be putting 2 cm cube of Benedict solution into each of the small test tubes with the concentration of the 10%, then we have the 1%, then 0.1%, then 0.01%, and at last we have the 0.001%. okay, so put the test tubes labeled 10 cm cube into the boiling water bath. Start the timing. okay, so now the 10 cm cube is actually the highest concentration, and now why we are actually putting the highest concentration, why are we actually timing it with the very first one. okay, so Actually, we time it for the very first, because I told you that the higher the concentration, the less the time is taken for the color change. Okay, but you do not have to do this. Okay, my suggestion will be that you start with the lowest concentration, because it will give you the most time. Okay, it will give you the most time, and it will actually give you the most time to react. Okay, so uh, measure the time taken for the first appearance of the color change in the test tube. If there is no color change after 120 seconds, stop timing and record more than 120. Record the result in the step 20, for step 26 in the A part 2. So actually we have to make the table. Obviously, remove the test tube from the water bath and put the test tube in the test tube rack. Repeat step 25 to 28 with the remaining concentration of the reducing sugar solution. So we have to do it with the other concentration like 1%, 0.1%, 0.01%, but I told you have to start from the lowest concentration. It will give you the most time to react. Okay, then we have to record the results in the appropriate table. So now let's make a table. Okay, so this is how your table should look like. Okay, we will actually have two columns. Okay, these will be your two columns, and then we will have 5 rows. Okay, the first one will be for heading, and what will be the heading? We will have concentration. Okay, because it is the independent variable and it is what we are changing. So concentration, I am just writing CONC because I do not have enough space. Okay, so concentration of reducing sugar solution. Okay, and what is the unit? The unit is percentage. And then the what is the other one? It is the time taken for first color change. And what will be the unit? You always have to take the time in seconds. Okay, and always in whole numbers, because the time stopwatch will actually give you something like this, 127.52. Okay, so what is 127? It is actually 2 minutes, or you can just say it is 2 minutes and 7 seconds. Okay, but you always have to take that in the whole number. For example, 127. Okay, like that only, not a decimal part of it. So, and now we will have 5 rows. Okay, so we will have 5 rows. So this is the row 1, then this will be the row 2, then this will be the row, let me just draw this again. So this will be your row 3, then this will be your row 4, and the last one is actually your row 5. Okay, so these will be your all of the rows. Okay, let me just correct this, and you have to make sure that the corner of the table are always closed. Okay, not something like this. Okay, like this, not like this, or you can say they should not have a gap, because the examiner deducts the marks for this. So then you have to just place the concentration in ascending order. For example, what was the smallest one? So it was 0.001%. Okay, now you do not have to mention the unit because you have already done that in the heading. So when we go down, we have to increase it, so it will be in ascending order. So this will be 0.01%, then 0.1%, then the 1%. Okay, you can just write 1.0, and then 10.0, which is the highest. So I told you that the highest concentration means the lowest time taken, and the lowest concentration means highest time taken. So let's just take more than 120 for this one. So more than 120 for this one, and let's just take this one as 115 seconds, this one as something about you can say let's just take it as 90 seconds. Okay, so it will be good. So 90 seconds, and the 1% as, let's take it as 60 seconds, and this one as 25 seconds. Okay, so this will be good. Let's just now you will be doing the test with the unknown solution, or you can say the one we actually performed the diffusion with. Okay, so now put 2 cm cube of Benedict solution B into a test tube P2, shake gently to mix, put this test tube into a boiling water bath and start timing. Okay, so measure the time taken, and also I want to give you one note that you have to put equal amount of the band aid solution and the solution that you want to test for. For example, if I'm testing for the solution P2, I have to add 2 cm cube of the band aid solution and 2 cm cube of the solution of P2. Then shake gently to mix. Okay, so put this test tube into a boiling water bath and start timing. Measure the time taken for the first appearance of the color change in the test tube. If there is no color change after 120 seconds, stop timing and record it as more than 120. Record the results from the step 32 in A part 3. Remove test tube from the water bath, put the test tubes in the test tube rack. Repeat step 30 to 34 with the test tube Q. So let's see what the 34 is. So it is actually, you have to test with the band solution with the Q2 as well. Now remember what was Q2? It was the beaker, the diffusion test with the beaker having 60 degrees Celsius water. Okay, so this means that the time will be taken less for the beaker with the 60 degrees Celsius, because the diffusion would be faster when the temperature is higher. So the temperature will be higher, so this means that diffusion will be faster, and more of the sugar content, or you can say the reducing sugar, more of the reducing sugar would be diffused. Okay, this is what it means. So let's take less time for the Q2, so let's just take it as about, you can say, 45 seconds. Okay, and then you have to for the P2, let's take it less. Okay, so let's just take it about as 125 seconds. Okay, so then using a result from A part 2 and A part 3, estimate the concentration. Okay, so now let's see that 125 for P2 and result for the Q2 is 45. So it will be according to what you test. Okay, so in the you can say in the lab. Okay, so if it is more than 125, this means it is more than 120. Let's just take a lower number. Okay, let's just take a lower number. Let's just take it as 105. Okay, so if I have 105, this means it is between 0.1 and 0.01. Now you can take any concentration between. Okay, so let's just take it as about, you can say, let's just take it as 0.05. Okay, it will be 0.05 percent concentration, so this will be 0.05 and percent, and then the other one is 45 seconds, so it will be between this one, the 1% and the 10%, but it is more closer to the 60 seconds, so this means it is more closer to that one. Okay, so let's just take it as about 2.0%. Okay, so let's just take it as 2.0%. So now explain the difference between your results for P2 and Q2. So now actually they are asking you that why the concentration of reducing sugar is in the P2 is low and why it is higher in Q2. Okay, you have to explain it. Now how we are going to be explaining it? We are going to be explaining with the help of with the temperature. Okay, so you will say that Q2 has higher temperature, so higher kinetic energy, which increases the rate of diffusion of sugar solution. Okay, hence the concentration of reducing sugar solution is higher. Okay, I just forgot the reducing here. So reducing sugar solution, and this will be your answer. Okay, state a dependent variable in the investigation you have just carried out. Now dependent variable is that the one that you are looking for, okay, the one that you are using to conclude your results, and it is fully dependent on your independent variable. Now what is the independent variable? The independent variable is that one that you are changing, and what we are changing here, we were changing the concentration. Okay, so your independent variable here was the one we were changing, which is the concentration. And what is being dependent on the independent variable is your dependent variable, which was the time, because the time was directly influenced by your concentration. The more the concentration, the lower the time. So it will be the time taken for first appearance of your color change. Okay, then identify one source of error in step 24 to 29. Suggest improvement to method which will reduce the effect of this error. Okay, so you have to look specifically to the range of steps they have given. So 24 to 29. So put 2 cm cube of Benedict solution B into each of the 5 small test tubes. Then shake gently to mix. Put the test tube labeled 10 cm cube into the boiling water and measure the time taken. And now what we are doing is that we are actually taking measuring the time taken for the first appearance of color change. And okay, so now this can be your potential error. Okay, the time taken for the color change, because you already know this error, that it will be difficulty to identify the color change. And what will be its improvement? Its improvement will be that you can use a colorimeter or you can use a color chart. Okay, so that the color changes, you can just identify it then and there. And then this can only be the error, I think. Okay, so you can also write other errors related to this experiment. Like for example, you can write that the outer side, or you can say the outer region of the whisking tube will be, you can say, it can be contaminated by other solution that will obscure your readings. Okay, and you can also write that when you take the solution from the external solution, or you can say the one that has been diffused to, so that external solution, if you that is actually not mixed well. Okay, it actually have unequal mixing. So because when it has been diffused to, so what will happen is that not every region will have the same sugar solution, so it will have unequal mixing. So what you can do, you can ensure that proper mixing happens, and then you can take a solution from that. But here we are being limited with the step 24 to 29. That is why I am just using this error, that difficulty to identify the color change. Okay, and what will be improvement? Use a colorimeter, or you can say a color chart. Okay, if you were not being restricted by the step 24 to 29, then in that case you could use the other improvement and error that I just told you. Okay, then suggest how you can modify the procedure to obtain a more accurate estimate of the reducing sugar solution in P2 and Q2. Now P2 and Q2 are actually your unknown solutions, and what we are doing here is that we are actually dealing with a very wide range of concentration. You can see that there is a very large difference by a factor of 10. So there is a very large difference between the concentration. Okay, so whenever there is a very large difference between the concentration, what you can just write about the modification is that you can use less wider you can keep less wider gap between the concentration by performing a simple dilution. Okay, then you will time each of the solution and you can just plot a graph, and then you can just intersect the graph at that point where your time of the unknown solution is occurring. Okay, so we will just write that. So use a wider gap between the concentration, and you also have to tell that how we are going to be keeping a wider less wider gap. Okay, it will be a less wider gap. Let me just write less and then wider. Less wider gap between the concentration by performing simple dilution. Then we have to time each solution, and you can also increase the accuracy of the experiment by repeating the number of times you actually calculate a time for each solution. Okay, you can take the average of that time. So time each solution, you can say 3 times or 4 times. Okay, it can be an improvement 3 times to reduce anomalies, and then at the end plot a graph for concentration and time, and use it to deduce the concentration of P2 and Q2 by checking the corresponding time. Okay, so this will be our answer. Perfect. Let's move on. Now we just have to make the graph. Okay, we are done with the experiment, so this is the easy part. So a student investigated the effect of concentration of the acid on the distance it diffused through the agar blocks, so containing the universal indicator over a period of 2 minutes. So they have given you, this is your independent variable and the other one is your dependent variable. Okay, so plot a graph of data in the table 1.3 on the grid in figure 1.4. Use sharp pencil for drawing graphs. Okay, so you just have to first of all calibrate the scale. You also have to mention the axis. So the x-axis is your concentration of hydrochloric acid and it is the units of mole per dm cube. Okay, so concentration of hydrochloric acid, and what is the units? It is mole per dm cube. Okay, then let's just reduce the size of this. Okay, so and then for the other one it will be distance acid diffused in 2 minutes. Okay, so it is the distance acid diffused in 2 minutes and units is mm. Now you have to check that how many uh, boxes you have been given. Okay, the maximum space that you can have between two calibrations on the scale is this much. Okay, these are 10 small boxes or two uh, 5 unit boxes. Okay, so this is the maximum. If you try to take more than this, If you try to calibrate more than this, then you lose marks. Okay, so your calibration should be present at a maximum of this much. Okay, so we have 1 2 3 4 5 big boxes, and what is the range? It is from 0.27 to 1. So if I just divide 1 by 5, so it will be 1 divided by 5, it will be 0.2. Okay, so perfect, it will be 0.2, and your scale should not be awkward. Okay, so 0.4, like it should not be something about 0.2175 like that. Okay, just take whole numbers like 0.2, 0.4, or you can say 0.1, 0.2, 0.5, or 0.1 like that. Okay, 0.6, then 0.8, and then 1.0. Okay, then also mention the second one, it is from it is actually till 9. Okay, so let's see how many boxes we have on this one. So 1 2 3 and 4 and then 5. Okay, so same you can take for that as well. So it will be 10, then it will be 8, then 6, then 4, then 2, and actually you are starting from 3. Okay, so if some of the people want to take you can say want to break the scale, you can do so. Okay, but here I'm just trying to keep it as simple. So now you just have to plot the points. So the first one is 1 and 9. So 1 and 9, so 9 will be something about this point. Okay, this will be 9, and we will have 1, so it will be something about, let's see, it will be this point. Okay, and then you have to draw a small circle around it. Okay, then for the next one, it is 0.89 and 8.5. So 8.5 will be something about this point, and 0.89 means this is 0.9, so just between this. Okay, so this point actually. So let's just see. So it will be about, let me just draw the point, it will be this point. Okay, let's just move it a little bit to this one. Okay, it will be like this. Okay, and this, then you have to draw a small circle around it. Okay, then again we have 0.75 and 4.5. Okay, so 4.5 and 0.75, so 0.75 will be between, this is 0.7. Okay, and this will be between here at this point, and we have 4.5, so this is actually 5, and this will be 4.5. Okay, so 4.5 and 7.5, so it will be something about you can say this point. Okay, so then you can just draw a circle around it. Okay, and then you will have 4 and 0.45, so 0.45 will be this point. Okay, and 4, so like this, and then it will be 0.27 and 3.8. So 0.27, this is actually 3, this is 0.28, so 27 will be between this. Okay, and it is, the other one is 3.8, so 3.8, this is actually 3, so 3.8 will be this point. Let me just point. Okay, so this will be the point, and then you just have, what you can do is that you can just also make a curve, but I would suggest that you can just uh, connect these points with the straight lines. Okay, so I'm just doing that right now. Okay, so these will be the points, let's just join them right now. Okay, so this is actually your last line, and this is how your graph will actually look like. Okay, so we have just finished this paper, and I have also told you about everything. You can just go into description, you can watch the whole playlist. For the question number 2, I have told everything about each of the single plant diagram, although I'm expecting something about to come out the plant. Okay, so the more usual, or you can say about the dicot stem. Okay, and also it is going to be something about plants. Okay, so because the examiner is not like testing nowadays the organs, something like the bronchus or something like that. Okay, so it is more usual to be um, about the plants, and you can just watch everything about the higher magnification drawing in that playlist. Okay, and you can also, I've also attached the link of the how you have to open the and fill the whisking tubing in the description down below, so go check in that. So I'll be trying to upload as many videos on this channel to make it the only hub where the papers get roasted before you do. Okay, so if you like this video, do share with anyone you think it will benefit. I'll be signing off now. So thanks for watching. I'll be meeting you in another video. Till then, take care, goodbye, and good luck for your paper.